Hi, my name is Naveed Ishaque. I'm a bioinformatics group leader at the Berlin Institute of Health and also associated with ELIXIR Germany. Today I'll be giving the introduction for a workshop titled Multitask Perturbation Modeling for Single-Cell Omics. This workshop was initially given at the ELIXIR All Hands meeting in 2024, and we're recording it once more for the ELIXIR Single-Cell Omics YouTube channel. So let's get started with cells. Cells are the fundamental unit of life. Together they form tissues and organs. Here we have a Lego representation of the brain. You can see at different parts of the brain there are different colors. These different colored Legos represent different cell types. These cell types have very unique functions. How can we determine the function of these different cells? We can do this by measuring their molecular profiles. However, there are different molecular profiles that we can measure. For example, we can measure their DNA, their RNA, their proteome and metabolome, and so forth. Here, in my talk, I will be focusing on measuring the RNA, or measuring the transcriptome. So whenever I refer to transcriptomics or gene expression, I'm talking about measuring RNA. For tissues and organs to function correctly, they need to have the correct cellular architecture, and those cells need to have the correct molecular profiles associated with their functions. Normal tissues can be pushed towards disease phenotypes through changes such as loss of important cells or changes in gene expression. This makes it very important for us to be able to profile the gene expression in different conditions for us to understand what's going on in normal cases and what's happening in different diseases. To better understand the cellular architecture and the gene expression programs active within cells in different conditions, we can perform transcriptome profiling. By and large, the majority of transcriptome data that has been generated so far has been based on bulk transcriptomics. This is where you take all of the cells and generate an average representation of the gene expression of every cell within your tissue. So as you can see on the bottom left, you don't get a very accurate representation of the cellular architecture, but you can see residual signatures of what was there in the brain. In the last decade, we've seen the rise and adoption of a new methodology called single-cell sequencing. So this is where you isolate individual cells from your tissues, and then you profile the gene expression of each and every cell. Once these have been measured, you can take computational approaches to identify cells that have similar gene expression profiles, and then cluster them together and identify changes in cell type architecture or gene expression patterns within individual cell types. So this new technology has enabled us to better understand the cellular architecture and the gene expression of those cells within tissues. Excitingly, in the last few years, we've seen the rise of spatial transcriptomics. This methodology is able to profile the transcriptome of individual cells in the native tissue context. So we know exactly where those cells were in the tissue slice that we analyze. So this brings about additional layers of information, allowing us to better resolve things such as how cells communicate with their neighbors to achieve higher order function within these tissues and organs. Parallel to advances in measuring the transcriptome of individual cells, there are a number of breakthrough technologies that have really advanced single-cell research. One of those technologies includes high-throughput screening. For example, one can perform genetic editing through CRISPR to see how particular gene knockouts can affect the function of cells. You can also apply high-throughput drug screening to see how these cells respond to external stimuli. Furthermore, advances in AI are now able to make use of the very large data that is generated by these single-cell transcriptome experiments to be able to identify nonlinear relationships that we have not been able to profile in the past. Combining single-cell profiling with perturbation modeling and advanced AI holds a lot of promise. For example, we can take tissues of interest, apply some sort of perturbation or some drug screening, and measure those outcomes. Those outcomes can be fed into advanced AI models where we can identify key genes that have changed through that perturbation. Or, in the case that we're doing drug screening, we can potentially identify effective drugs against particular diseases. We're only at the initial phases of exploiting these technologies, and hopefully in the subsequent presentations, we can demonstrate how they can all work together to better understand biology. Thank you for your time. I will now hand over to George, who will tell us a little bit more about perturbation modeling. Thank you.